Hi everyone, I'm Amy Goldman from Pacific Northwest National Laboratory. I want to start by saying that a subset of samples for this project were collected in the Yakima River Basin in Washington State. We acknowledge the Yakima Nation as owners and caretakers of the lands where we collected those data. We thank the Confederated Tribes and Bands of the Yakima Nation Tribal Council and Yakima Nation Fisheries for working with us to facilitate sample collection and optimization of data usage according to their values and worldview. Additionally, this project is done in partnership with Stefan Gary at Parallel Works, and it's funded by the DOE Office of Biological and Environmental Research Environmental Systems Science Program, and by the DOE Small Business Innovation Research Program. There are captions available for this presentation at the QR code on the slide or at tinyurl.com/Goldman-AGU22. I'm excited to talk with you about what we call the ICON-ModEx sampling campaign, which iterates between artificial intelligence models and field sampling to study river corridor biogeochemistry. I will get into more detail at the end, but I want to mention up front that this campaign is still ongoing and that you can get involved in the sampling or in other aspects of the campaign. I also encourage you to think about how you might be able to apply pieces of this approach to your own research. I use the term ICON-ModEx, so I want to start with answering the questions: what is ICON science, why are we using it here, and more broadly, what can it do for you? ICON stands for Integrated, Coordinated, Open, and Networked. It's a framework to guide the intentional design of any project motivated by the pursuit of mutual benefit and transferable knowledge. In scientific training, we're not often taught to approach the entire design of our work with intentionality. The emphasis is usually on the desired scientific outcome. So here we're thinking about these principles from the very start of our ideation and carrying them through the research lifecycle. So for I in ICON, we are talking about intentionally integrating across disciplines and scales by design, so more perspectives, connections, and analyses are planned for and optimized. For C, this is intentionally coordinating with consistent protocols and methods to enable transferability. This is not simply having interoperable data. This is thinking about how those data were actually produced to create information that can be combined down the line. The O is for intentionally open and fair throughout the research lifecycle, from ideation to publication. This is supportive, of course, of openly publishing your data or manuscripts at the end of a project, and it's also thinking about what can be done beyond that to be open much earlier and more creatively. And then N is being intentionally networked, whereby research is designed and or implemented with a broad range of stakeholders to ensure mutual benefit. And we use the term stakeholder here broadly to refer to any individual, team, institution, community, or other entity that may be interested in or impacted by the work. But I want to acknowledge that some of the folks we're working with are rights holders. With this networked principle, we're making choices based on understanding what other people value beyond our core research team. This involves taking time to learn from people and sincerely making changes to meet the needs of others while staying true to the project's scientific needs. We're going to dive in now to the ICON-ModEx campaign, which is one example of ICON science in practice. As many of you know, hypereic zones have been shown to contribute approximately 4 to 96% of stream respiration, which is nearly the largest range possible. There's also no model to help us predict or understand this variability. The goal of this campaign is to predict hypereic zone respiration, including hotspots, and also to understand molecular to continental scale controls. The field portion of this work is being done through the Worldwide Hydrobiogeochemistry Observation Network for Dynamic River Systems, or WHONDRS, which is a consortium formed by the scientific community studying river corridor hydrology, biogeochemistry, and microbiology. The premise of this campaign is based on a model experiment iteration cycle, or ModEx. The process here began by using open access data to build a collection of artificial intelligence models to predict sediment respiration. Those predictions were used to guide the selection of sampling locations to collect more data. Samples are now being collected every month via WHONDRS crowdsourcing, and those samples are sent back to us at PNNL for data generation to feed back into the model. The new data generated monthly are used to test and improve the collection of AI models, and those results are in turn used to guide sampling locations for the next iteration. In practice, this means that the AI models generate a list of priority sampling sites. So one example of why sites are high priority is that they are outside the previously sampled environmental conditions, thereby pushing the bounds of the models. So on the left, we have the highest priority sites from the first model output, and then after we collected samples and used the new data to remake the models, we generated the map on the right as the next iteration of highest priority sites. You can see that from a single ModEx cycle, the sampling guidance has shifted, particularly along the eastern coast of the US. When we look at how repeated ModEx iteration is affecting the predictive capability of the models, we see an increase in the fraction of respiration explained by the models as the number of ModEx cycles has increased, though the uncertainty in how much variation is explained has also increased. There are many more steps to make monthly contiguous US scale ModEx possible. Every month, we receive samples, generate new AI models, and generate a new list of priority sampling locations. Then, we send out a map of those locations to potential collaborators. This sampling is open to anyone in the contiguous United States. Collaborators sign up to sample 2 months in the future, so the map generated in December will be used to sample in February. We work with the collaborators and our lab team to confirm the scheduling and send out a sampling kit to the collaborator. The collaborators then use the kit to collect samples and briefly deploy a dissolved oxygen sensor. And then we repeat this ModEx cycle monthly. The idea is to continue until we see no more gains in predictive capacity. To link this back to ICON and to make this more tangible if you're interested in pursuing something similar, this campaign integrates across physical, chemical, and biological data and AI models, coordinates by sending out free sampling kits to collaborators and generating and publishing data according to consistent protocols. We also began this campaign with open study design discussions that were widely advertised, and then in linking to the networked principle, we used feedback from those discussions to make changes to the study design to better meet the needs of stakeholders. We're also publishing the data openly and fairly prior to using it for our own analyses and putting the AI models on GitHub for public access. And as another component of Networked, we've begun doing classroom presentations about the campaign after we received feedback from stakeholders that they were interested in that type of engagement. As I mentioned at the start of this presentation, this campaign is ongoing and there are many different ways people can get involved. You can use this QR code on the slide to sign up for more information. Anyone in the contiguous US can reach out about collecting data and samples at a field site of interest to you. There are also many possibilities for just picking up the data produced and advancing science leveraging this campaign. We're also interested in partnering on proposals, presenting in classrooms, and more. So please reach out to us if you're interested. Thank you.